If you watch a dog sleep for long enough, you will eventually see it. The closed eyelids darting back and forth, the muffled closed-mouth vocalizations, the paws rapidly paddling against the floorboards. It is common to dismiss this as mere neurological noise, a random muscle spasm or a meaningless misfire of the nervous system as the body powers down. These movements are measurable artifacts of rapid eye movement, or REM sleep, a brain state characterized by intense neural activity. This process involves episodic memory, which is the brain's internal record of specific chronological and spatial events experienced during waking hours. Researchers have observed a correlation between these physical twitches and the neural signatures used to process those episodic memories. We rarely see this in healthy adult humans, who remain largely motionless during REM. Canines, however, offer a highly visible display of the dreaming brain at work. By placing non-invasive electrical sensors along a dog's scalp and muscles, researchers can capture the exact neurological data driving these physical movements. This biological quirk provides an observable opportunity to map out how mammalian neuroanatomy processes information. The familiar twitch on the living room floor is a visible data leak. It is the external evidence of an internal, high-speed memory sorting process. To understand why that data leaks out as physical movement, we first have to map the electrical architecture of the canine sleep cycle. The cycle begins with slow-wave sleep. This is the initial deep rest phase that immediately follows wakefulness. On this graph, the electrical signature of slow-wave sleep drops from the waking baseline into slow, high-amplitude waves. Then, physiology spikes abruptly into jagged, mixed-frequency patterns of REM sleep, indistinguishable from a waking brain. Dogs hit this intense REM phase fast, usually within 20 minutes. This makes up a total canine sleep cycle of about 45 minutes, exactly half the length of an extended 90-minute human sleep cycle. During this REM phase, the canine cortex is computationally acting as if the animal is awake, generating motor commands, processing spatial data, and firing visual stimuli. Because the canine brain plunges into this highly active, pseudo-waking state so quickly, it requires an equally rapid systemic shutoff valve to stop the dog from physically running into walls or injuring itself. That shutoff valve is managed by an anatomical control center located deep in the lower brainstem. Its function is to induce muscle atonia. This is the active chemical paralysis of the skeletal muscles during sleep. This paralysis originates in a brainstem cell cluster called the pontine tegmentum. During REM, it projects inhibitory neurotransmitters straight down the spinal cord. These chemicals form a barrier at the motor neurons, blocking movement commands from reaching the legs or tail. But in canines, this inhibitory barricade is naturally imperfect. This imperfection results in motor leakage. The chemical paralysis fails to block 100% of the outgoing electrical signals, allowing fragments of motor commands to slip past the brainstem. We see this motor leakage most prominently in puppies and juvenile mammals, whose inhibitory pathways and nervous systems have not yet fully developed the capacity for total lockdown. When you observe those twitches on the floor, you are seeing the precise moment when the chemical paralysis fails to completely contain the electrical force of its dreaming brain. If we follow those leaked signals back up the spinal cord and into the cerebral cortex, we can identify exactly what commands are breaking through the barricade. This points us to the hippocampus, the core engine responsible for memory retrieval during the REM cycle. While the dog dreams, the hippocampus locks into a continuous data loop with the motor cortex, feeding it spatial and behavioral information. Empirical tracking of this data loop reveals that the leaked motor commands are highly organized sequences. They are not random firings of a resting brain. When researchers map the specific rhythms of these twitches, the data matches them perfectly onto recognizable waking behaviors. The dream signatures are often entirely breed-specific. Pointers exhibit their distinct stance. Terriers fire off rapid digging sequences, and retrievers rhythmically move their jaws. This process is known as procedural memory encoding. By replaying these exact motor sequences, the brain learns, refines, and perfects physical skills without the body ever moving. Firing these sequences repeatedly during sleep physically strengthens the synaptic pathways, ensuring the dog is faster and more coordinated in waking life. The hippocampal replay acts as a necessary high-speed data transfer, shifting newly acquired short-term experiences into deeply rooted long-term instincts. This repetition during REM sleep integrates the day's spatial and motor lessons into the nervous system, turning temporary experiences into permanent traits. This canine neurological model extends beyond a single species, shedding light on the wider mammalian family tree. Through comparative neurobiology, researchers use the observable traits in one species to understand the fundamental wiring of another. The exact mechanisms of hippocampal memory replay and muscle atonia we observe in dogs are evolutionarily homologous to the architecture of human sleep. This comparative data shows that the strict correlation between REM sleep percentages and post-sleep memory retention remains remarkably consistent across species. In fact, the leaky atonia seen in juvenile mammals likely serves as an evolutionary feature, allowing young animals to safely and physically practice critical survival skills while remaining hidden and asleep in the den. Mapping this canine sleep architecture also provides a vital, non-invasive baseline for studying human neurology. It gives us a model for understanding REM behavior disorders and tracing the early stages of memory degradation. When we return to the subtle twitch of a sleeping dog, the neurological data entirely recontextualizes the phenomenon. An active neural state triggers a partial failure in chemical atonia, which in turn reveals a physical playback of the day's memories. These internal simulations serve a specific physiological function, ensuring the brain can organize complex data efficiently. Observing a dog dream provides a direct look at the biological architecture that enables mammals to consolidate memory and navigate their environment.